A very good evening, everyone. So we'll just wait for 2-3 minutes for everybody to join and then we'll start the session. वॉशरूम होके आते हैं। So in today's class, we'll be covering the questions which are related to week 10. So I'll just wait for one more minute and then we'll start the class, okay? Okay, so let us start today's class. So, this week we'll be covering the questions from week 10 of our course. So, in the previous class, we have started with plant developmental biology, and in this class, also, we'll continue questions related to plant developmental biology only. And subsequently, we'll shift to the animal part. So, first, we'll deal some questions which are related to plant developmental biology, and then we'll go to the animal developmental biology part. So, as we always do, I'll give the question. I'll give you 2 to 3 minutes to solve it, and then we'll discuss it. Okay. So, this is how we'll proceed. So, let us quickly see our first question. So, the first question says, in the image shown, which part will eventually give rise to organs involved in fertilization? So, you can see there is an image, and you have various parts which are labeled as A, B, C, and D. So you have to tell that which organ eventually will be involved in the fertilization. Okay, so first of all, you should know whether it's a part of a plant or an animal because it doesn't really describes it here. And then you have to identify that which part among the following parts which are labeled will eventually be involved in fertilization. Okay, so I'll give you 2 minutes. After 2 minutes, we'll switch the screen and we'll discuss it. 1 minute more. Okay. So now I'll switch the screen and we'll discuss it. Let us see. Okay. So the question says, in the image shown, which part will eventually give rise to organs involved in fertilization? So this image that you can see on the screen, it's actually a floral meristem in longitudinal section. So this is the image of a floral meristem. Okay. And this is a longitudinal section of a floral meristem. So this A part, these are the outer walls, which are basically your sepals or petals. Okay. This B part is the central primordium, which will eventually develop into stamen or carpels, which are the reproductive organs which are involved in fertilization. The C part is the petal primordia, and the D part is the sepal primordia. Okay. So this is sepal primordia. This is this C part. I'm writing it over here. This is the petal primordia, and the B part is actually the central primordium, and this B part will eventually give rise to stamen and carpels, and stamen and carpel are the reproductive organs which are involved in fertilization. Okay, so this B part, which is the central primordium, this is actually uh, the part which develops into stamens and carpels, which are the reproductive organs. Okay, so the correct answer would be B. So B is the part which eventually will give rise to stamen and carpel, which is involved in fertilization. Okay. Next question is, the plant hormone dash is important for maintaining dormancy, whereas the plant hormone dash is required to break the dormancy. So you have to name two plant hormones. The first one, A, which is required for maintaining the dormancy, and second, B, which is required to break the dormancy. Okay, so these are two questions which I have clubbed into one. So there are two parts of this question, A and B. So you have to name two plant hormones. So it's very easy. I think you must all have already studied in your previous classes also. But I'll also tell you the mechanism by which these hormones maintain or break the dormancy. But first, I'll give you 2 minutes. After that, we'll discuss the answer. Okay. Think, you have 2 minutes to think, and then we'll discuss the answer for this question. Okay. 1 minute more. 1 minute more. Okay, so let us discuss this question. So, the plant hormone dash is important for maintaining dormancy. So, which hormone is required for maintaining dormancy? It is abscisic acid. So, abscisic acid is the plant hormone which is required for maintaining the dormancy. So, this is the answer for the A. The B is the plant hormone which is required to break the dormancy. So, which plant hormone is required to break the dormancy? It is gibberellins. This is required to break the dormancy. Okay. So, now let us see how abscisic acid is maintaining the dormancy. So basically, what it does is that it prevents the premature germination of the bud. So basically, it helps the seed to tolerate the drying environment and keeps the metabolic activity of the seed very low. So, because it is preventing the premature germination, so in seeds, when there is high abscisic acid level, it is basically during the seed development phase. It induces storage and protein accumulation and also some kind of desiccation tolerance proteins, which we call as LEA proteins, which repress the germination programs. Okay, so what happens at the time of germination? Gibberellic acid, it basically what it does is that it activates some kinds of enzymes and gene programs that mobilizes the stored reserves and permits the embryo to grow out. Okay, so if we talk about abscisic acid, so we write it in short form as ABA. So it is actually preventing the, it prevents abscisic acid, it prevents premature germination, premature germination, and it helps seed to tolerate the drying environment, and it keeps the metabolic activity low. Okay, now so when the seed is actually developing, so at the time of the development of seed we do not want it to germinate prematurely. So at that time excessive abscisic acid is playing its role. It helps to keep the seed intact and does not allow any kind of premature germination. Once the seed is fully developed and it is now ready for germination, at that time the levels of abscisic acid has to reduce, and at that time gibberellins comes and does it function. So what it do is that it activates the enzymes that helps in the germination of the seed, which helps in breaking the seed dormancy. So what it does it, that gibberellic acid which is present in the embryo, it is produced in the embryo, it signals some kinds of tissues to make alpha amylase and other kind of hydrolases enzyme. And these enzymes, they actually break down the starch into sugars, which is important to fuel the embryo. So this is what gibberellins do. So gibberellins, in short, we write it as GA. Okay. So it activates enzymes. Gibberellins activates enzymes and gene programs that mobilizes the stored reserves of nutrients. Okay, that mobilizes stored reserves and permit the embryo to grow out. The embryo to grow out and break the dormancy. So I hope this question is clear to you. Fine. Now the next question is, which of the following tissues are responsible for increasing the stem girth? Okay, I'll just label it as A, B, C, D, and E. Fine. Now the question is, which of the following tissues are responsible for increasing the stem girth? A. Cork cambium. B. Vascular cambium. C. Intercalary meristem. D. Shoot apical meristem. And E. Root apical meristem. Okay. So girth means the thickness. So which tissue is responsible for increasing the thickness or the girth of the stem? So uh, there can be more than one tissue which is involved. So there can be more than one correct option for this question. So I'll give you 2 minutes and then I'll switch the screen and just a minute. Just have to see something. Yeah. Okay. Fine. So this is the question. You can have more than one correct answer for this question. Go through all the options. I mean, you are able to, you can answer this question if you know the function of each and every tissue. So we'll discuss it in detail. So try to answer this question. I'll switch the screen. We'll discuss after 2 minutes. Just keep in mind that girth, we are talking about, when I'm talking about girth, it means the thickness of the stem. Okay, so let us discuss this question. So the question says, which of the following tissues are responsible for increasing stem girth? Okay, so the correct answers are, there are two correct answers for this question. The first one is vascular cambium and the second one is cork cambium. So why vascular cambium? Because vascular cambium basically it is the primary driver of secondary growth. That means it helps in increasing the thickness of the stem. So it produces the secondary xylem on the inside and secondary phloem on the outside, which adds the bulk to the stem's thickness. Okay, so therefore vascular cambium is one answer. Second is cork cambium. Why cork cambium? Because basically cork cambium it is formed during the secondary growth stage, usually in the older stems, and it produces cork outward and secondary cortex inwards. And though its main role is in the protection, that is in the periderm formation, but the addition of new cork cells, it also contributes slightly to the increase of the girth. Okay, so the two answers would be vascular cambium and cork cambium. Why vascular cambium? Because it is the primary driver of secondary growth. Okay, it is primarily it is involved in the secondary growth. When we talk about secondary growth, we are talking about increase of the thickness. So when we talk about stem girth, we are actually talking about the thickness of the stem. Okay, and it adds to increasing the thickness of the stem. And this cork cambium is basically uh, main role of cork cambium is protection, but because it is adding new cork cells, that then it is also contributing in the thickness. So now the other options such as intercalary meristem, shoot apical meristem, root apical meristem, why are they not the correct answers? See, intercalary meristem it helps in elongation and not thickening. Okay, so this is involved in elongation, not thickening. Then your shoot apical meristem, it is also involved in increasing the length. Again, it is involved in increasing length. This root apical meristem, it basically it is a root meristem, so it is involved in elongating root and not the girth. So elongates roots. And this one cork cambium, basically it is involved in protection, but as it adds new cork cells, so addition of new cork cells, it also contributes to the girth increase. Okay, so I hope this question is clear to everybody. Okay, let us move. So the correct answer is cork and vascular cambium. Okay, now this is a little, it might look a little confusing. The question is to match the proteins with their roles. So we have constants, terminal flower 1, which we call it as TFL1, Aptella 1, Superman, AP3 on one side. On the other side, we have region specifying gene, vegetal meristem to inflorescence meristem transition, floral meristem identity gene, floral induction gene, and floral organ identity gene. So I'll give you 3 minutes. In 3 minutes, you try to answer this question and then we'll discuss it. There may or may not be more than one correct answers. You have to see all the options. You try to make all the possible pairing that can be done, and then you try to look at the options which are given to you. Okay, so you have to match according to the options which are provided. There can be more than correct answer or a better answer, but if it is not given in the options provided, then it is of no use. So you have to match it according to the options which are provided to you. So in order to answer this question, you should know the role of the proteins. Aptella, AP3, terminal flower 1, constants. Think where they are involved, in which pathway or in which process they are involved in. Can you think of the options? Okay, let us try to solve this question now. So the first protein is constants. Okay, in short, we write it as CO. So constants is nothing but a floral induction gene. Okay, so it is a photoperiod pathway regulator that promotes flowering. That means it induces the flowering patterns. So constans is nothing but a photoperiod pathway regulator. It's a floral induction gene. So according to this, which uh, one matches with this given information, so we can say with one, floral induction gene is an option, so it matches with the D option. So we can say that one matches with D. Second is TFL1, which is terminal flower 1. So what is TFL1? So TFL1 is basically inflorescence. It is involved in inflorescence meristem transition. So what TFL1 does is that it controls the inflorescence meristem identity and the timing or transition to flowering. Okay. So what happens that if there is a loss of this particular gene, if there is a loss of TFL1 gene, which is basically a terminal flower gene, then what happens is that there is a it causes the loss of a terminal flower. So I hope this is clear to you. Then according to the information provided, which option will go the most with this question? It is B. That is floral, vegetal meristem to inflorescence meristem transition. So second one goes with B option. Then what is the third one? Third one is epitella. So epitella 1. So what should be the answer for epitella 1? Basically, it is a floral meristem identity gene. Epitella 1 is floral meristem identity gene. Basically, it is involved in the early flower development and specifying the floral meristem identity of a plant. So, according to the information provided, this will go with option number. Which option it will go? It will go with option number C. So, third is C. Fine. Next is Superman. So, this Superman, what is the function of this particular gene? It is a region specifying gene. So, this Superman is basically a region specifying gene, and what it does is that it defines the boundary or the regions between stamen and carpels. Okay, this gene defines the boundary between stamens and the carpels. So ultimately, this gene is required in specifying the domains. So it is needed for domain specification. So which option it goes? It goes with option number A, which is region specifying gene. And the last one is AP3, which is epitella 3. So epitella 3, obviously the last which is left is option number E, which is floral organ identity gene. It is floral floral organ identity gene. Okay. So with this information, what should be the answer? The answer would be this one. So please match it with whatever we have discussed. Is it following the concepts that we have discussed just now? Just check it. Check it once. First D, second B, third C, fourth A, and fifth E. Fine. Okay. So let us move to the next question. The next question says, scientists have recently discovered a plant species that will bloom flowers only after surviving a winter period. Okay. Which of the following pathways may be a dominant flowering pathway in this plant? Your options are gibberellin pathway, autonomous pathway, photoperiodic pathway, vernalization pathway, and auxotrophic pathway. 2 minutes and then I'll switch the screen. Try to solve it. think about all the options. Just label the options as A, B, C, D so that you can answer them. 2 minutes to solve this question. 1 minute more. Think. So the question is saying that a plant species that will bloom flowers only after surviving a winter period. So that means that these flowers require a winter period also, isn't it? That is what the question is saying. So, which kind of pathway is involved in such kind of flowering plants? That you have to tell. Okay, so I'm switching the screen. Let us discuss the question then. So, if a plant blooms only after experiencing a winter period, it means it requires exposure to prolonged cold to trigger the flowering. And this process is known as vernalization. Okay, so what is vernalization pathway? Vernalization pathway ensures the flowering only after the plant has gone through winter, preventing premature flowering in autumn season. Okay, and genes such as FLC, which is known as flowering locus C, which is basically a floral repressor, these genes are silenced by vernalization, and this allows flowering genes like FT, SOC1 to be expressed in the spring. Is it clear? So just remember that these plants flower only when they have gone through a winter season. So there is no premature flowering in the autumn season. And on the molecular basis, what it does it that genes such as flowering locus C, FLC, which is a floral repressor, these genes are silenced by the vernalization process, which allows the flowering genes like FT, SOC1 to be expressed in the spring season. Is it clear to everybody? I hope this is clear. So the correct answer for this question is vernalization pathway. Okay? Yes. Now, next question is how many cells are present in a human embryo? So, up till now, we were discussing the questions which were related to plants. Now, this is a question. We might have one or two questions which are related to plants even now, but this question is basically from the animal part of the developmental biology that we're dealing with. The question is how many cells are present in a human embryo at the morula stage? It's a very simple question, but while we are solving this question, we'll try to discuss all the stages so that you have a clear picture. Okay? You have 2 minutes to answer this question. In order to solve this question, which you should first know that what is a morula stage, which stage is known as a morula stage. One more minute left. Think. So the correct answer is 16. Okay. Now, can you tell me which stage is the morula stage when we talk about a human development, embryo development? Have you any idea? Think. This is an important concept. So you must know the developmental whole developmental stage for this question. Okay. So just give me 2 minutes. Meanwhile, you can think for the answer. Okay. So now, morula stage of a human embryo is reached after several cleavage divisions of the zygote. Okay. So morula is a solid ball of blastomeres which resembles like a mulberry. And because this morula resembles like a mulberry, therefore the name is morula. And in humans, the morula stage typically appears at around 16 to 13 to 32 cells division. So generally we call it as 16 cell division. Approximately 3 to 4 days after the fertilization, before the blastocyst is formed. This you have to remember. Okay. Before the blastocyst is formed, this morula stage is at around 16 to 32 cell stage, and it is basically a solid ball of blastomeres. This morula. Fine. So now we'll discuss in detail the whole process of development. Uh, just give me 2 minutes. I'm also doing some work. So in 2 minutes, I'll get free and I'll tell you about this. Okay. Sorry for the interruption. I had some work. Uh, that's why I had to go in between. Anyways, so now, uh, so we'll be discussing the stages. So day zero is actually the fertilization stage. Okay. So your day zero, it is the fertilization stage. So what happens? Basically, the male sperm and the female oocyte, it fuses and it forms a zygote, which is 2n, zygote, which is 2n, and which is totipotent. And then you come to day 1, which is a two cell state. Okay, it's a two cell stage. And what happens on day 1? First cleavage occurs, that is, the first mitotic division occurs without any growth, and two equal blastomeres are formed. And it is still enclosed in the zona pellucida, that is, outer glycoprotein coat. This is your day 1. Then comes your day 2. Day 2 is a four cell stage. Okay, it's a four cell stage. What happens in day 2? Further cleavage occurs and four blastomeres are formed, and each blastomere is still totipotent. So here it is the zygote which is formed on day 0. It is totipotent, obviously. Day 2, still it is totipotent. Then we come to day 3. Day 3 is basically a 8 cell stage. Okay. Which is, what happens here is compaction. So by this stage, blastomeres undergoes compaction and cells adheres tightly, forming a more solid like a ball. And it is a beginning of cell polarity and specialization. So from day 3, it's a beginning of cell polarity and specialization. Then your day approximately 3 to 4, we can say. Here comes the stage that we are talking about, which is the morula stage, 16 to 32 cells. Okay, and here multiple cleavage, it produces morula, and this morula enters the uterus, and cells are enclosed in zona pellucida. So this morula basically enters the uterus, and then comes your day 4 to 5 approximately, and this day 4 to 5 is your early blastocyst. Okay, early blastocyst. And blastocyst is nothing but a fluid filled cavity which forms inside the morula. Okay, it's a fluid filled cavity. Fluid filled cavity inside the morula. Okay. And then zona pellucida is still intact. And then comes your day 5 to 6. Day 5 to 6, where there is hatching. It is a late blastocyst stage. Hatching occurs. And then day 6 to 7 is the beginning of implantation. So these are all the stages. Okay, fine. Let us come to the next question quickly. Next question is, which of the following embryonic parts prevent the implantation of the embryo in the fallopian tube? Please note, here we're talking about prevention of implantation in the fallopian tube. Your options are A, cell membrane, B, zona pellucida, C, blastocoel, D, corona radiata, and E, vitelline membrane. Okay, here we are talking about prevention. That is an important thing to just see here. Try to answer this question. You have 2 minutes. Please please note we're talking about prevention of the implantation and not in the uterus, in the fallopian tube. Okay, that you have to pay attention to. Okay, so let us discuss this question. So, the correct answer for this question is zona pellucida, that is option number B. Okay. So why zona pellucida is a correct answer? See, zona pellucida is a glycoprotein coat which surrounds the mammalian oocytes and early embryo, and it remains intact up to the blastocyst stage. Okay, and it prevents the premature implantation of the embryo in the fallopian tube. And this premature implantation in the fallopian tube, this is termed as ectopic pregnancy. And it also prevents the polyspermy. What is polyspermy? Entry of multiple sperms at fertilization. So it has dual functions. So it prevents the premature implantation of the embryo in the fallopian tube also, and it prevents polyspermy also. So the embryo, it hatches out of the zona pellucida only after reaching the uterus around day 5 to 6, allowing the implantation at the right stage. Okay, so therefore the correct answer is zona pellucida. Now, why other options are incorrect? Cell membrane. What is the function of cell membrane? Basically, cell membrane is just a plasma membrane which has no role in preventing your ectopic implantation. Ectopic implantation is the premature implantation in the fallopian tube. Okay. Blastocyst is a fluid filled cavity inside the blastocyst. It is not related to implantation prevention. Then corona radiata, it is a layer of granulosa cells around the ovum, and it is lost soon after the fertilization. So it is again no role. And vitelline membrane, basically it is a membrane which is found in non-mammalian eggs like amphibians, birds. So it is not present in mammals or humans. So it has no role at all. So the correct answer is zona pellucida. So I hope this question is clear to everybody. Okay. Next question is, which of the following are necessary to maintain the stemness of the embryo? OCT4, okay, I'll just label it as A, OCT4, B, NANOG, C, CDX2, D, STAT3, and E, GATA6. Okay, which of the following are necessary for maintaining the stemness? Think what can be the answer. One more minute left. Okay. So now the correct answers are Oct4, Nanog, and Stat3. Okay. So Oct4, why Oct4? Oct4 is also known as POW-F51. So basically, it is a master regulator of pluripotency. It keeps the embryonic cells undifferentiated and it works with SOX2 and nano. Okay. Then nano, it is critical for self-renewal and maintaining the pluripotency, and it prevents the differentiation into specific lineages. And STAT3, it is involved in LIF, which is leukemia inhibitory factor signaling pathway, which supports self-renewal of embryonic cells, especially in mice. So why not CDX2 and GATA6 are involved? Why they are not involved in stemness? Basically, these CDX2 and GATA6, they are differentiation factors. They are not for stemness. They are not required for pluripotency or stemness. They are differentiation factor. So CDX2 is required for promoting profectoderm fate. Okay, and GATA6 is required for promoting primitive endoderm differentiation. So they are not the factors which are required for maintaining the stemness of the embryo. Okay. Okay, now this is an important question. In the image shown is the wild type expression patterns of the ABC class protein as per the ABC model for floral organ specification. Scientists discovered a mutant that does not form petals but can still form sepals. What are the possible expression patterns of the ABC class proteins in this mutant plant? And these are your options. Now just see, when only A is present, then it forms sepal. When A and B are present, it forms petal. When B and C are present, it forms stamen. When only C is present, it forms carpel. So the question says scientists discovered a mutant that does not form petals but can still form sepals. That means some problem is there. In this portion, there is some problem. What are the possible expression patterns of the ABC class protein in this mutant plant? Now, it is a very logical question. You see, even if you have no idea about the ABC model of plant, which eventually, anyways, we are going to discuss, but I'm just telling you that even if you have no idea about this ABC model, even then, with your logical reasoning, you should be able to solve this question. It's a very easy and logical question. For petal formation, you require AB. For sepal formation, you require only A. There is a problem in petal formation, that is, this should be right, this combination should be wrong. So you just have to select a combination where this is wrong but this is right. You understand? It's a very logical question. Think. Anyways, we are going to discuss it. Just think about the question. One more minute left. Okay. See, from this question, we can say that petals are formed when B and A both are present. Okay. And for sepals, only A is required. So let us first look for sepals. Can this form sepal? And these are the roles: role 1, role 2, role 3, and role 4. Role 1 is basically responsible for forming the sepals. Here, only A is present, sepals can form. Only A is present, sepals can form. A and B are present, sepals cannot form. Only A is present, sepals can form. C is present, A is not present, sepals cannot form. So, these two options are already cut. Now let us look at this option. We want option where sepals are forming but petals cannot form. So for petals we need B and A. Here B and C. Can it form petals? No. Here A is present. Will it form petal? No. Here this is the already incorrect. Here B and A present, so it will form petal. But we need the ones which cannot form petal. So this is again we cannot. This is already there. So from here we get the correct answers are A and B. Easy, no? The logical reasoning itself is able to explain this question. But let us also quickly see the ABC model of flower development. Okay? Because this ABC model of flower development is actually a very important topic. And as I try, like in my all my classes, I always try to include some of the topics which are not only important for your course per se, but which are also important for some of your competitive examinations. Let's say CSIR or DBT or UGC, whatever examination you are giving to. So this ABC model of flower development is a very important topic in plant developmental biology, plant physiology, and questions comes from this topic in your competitive examination. So if you are planning for higher studies, if you're planning for PhD or even for some MSc examination, you should have a very good grasp and idea on this ABC model of flower development. So, I mean, we cannot cover the every aspect of this ABC model of flower development, but I'll tell you a gist which will make base for you so that you can go at home and you can just have a more like a detailed uh, understanding of this topic. So this ABC model of flower development, it explains how different floral organs. What are different floral organs? These are sepals, petals, stamen, and carpels. How different floral organs are specified in plants, like, for example, for Arabidopsis thaliana. So there are four roles which are present in flowers, and these are concentric roles. First is sepals. First role, the outermost, is the sepal. Then you have petals. Then you have stamen, and then you have carpel. So role 1 is sepals. Role 2 is petal. Role 3 is stamen, and role 4 is carpel. And there are three genes, A, B, and C. And the combination of these genes will decide that whether sepal will be formed, petal will be formed, stamen will be formed, or carpel will be formed. So these A, B, C are the uh, three classes of genes, and these are called as Matchbox transcription factors. Okay. Sorry. Matchbox transcription factor. So A class of gene, when we talk about A class of gene, so it codes for epitella 1 and epitella 2. Epitella 1 and epitella 2. And B class of genes, it codes for epitella 3 and pistillata. Okay, pistillata. And C class of genes, it codes for agamus. And other also, but just for example, agamus. So for sepal formation, only A is needed. Okay, sepals is formed only when A is there. That means only when A class of genes are present, it forms sepal. When both A and B class of genes are present, then it forms petals. When both B and C class of genes are present in role 3, then it forms stamens. And when only C class of genes are present, then it forms carpel. Is it clear? For sepal formation, you require only A class of genes. For petal formation, you require A and B both classes of genes. For stamen formation, you require B and C both classes of genes. And for carpel formation, you require only C class of genes. So these are the three classes of genes, and these are the examples of genes which comes under these classes. So this is what is needed. Now, let us see, there are three mutation conditions. When there is a mutation in the A gene, so now A gene is mutated. So when gene is mutated, C comes into its place. When C gene is mutated, A comes into its place. This is a rule. So when A is mutated, then C will come into its place. So since only C is there, it will form carpel. Here A is mutated, so only A is mutated, so here it will come C. So C plus B, it will form stamen and carpel. So it will be carpel, stamen, stamen. So when A is mutated, always remember, when A is mutated, then C comes into its place. And when C is mutated, then A comes into its place. So here, wherever there was A, everywhere C will come. When B is mutated, nothing can take over its seat. So A sepal will be formed. Here A plus B requires petals, but since B is mutated, B is not there, so only A is present. So again sepal will be formed. Here B plus C, but B is not there, so only C leads to carpel, carpel. Sepal, sepal, carpel, carpel. And when C is mutated, so when C is mutated, what will happen? A will take over. So here normal, sepal, petal. Here it should be B plus C, but it is A plus B, so it will form petal. And here it should be only C in normal condition, but since C is mutated and A has taken over its place, so it becomes sepal. So this is very important, guys. Okay, that's why I have included it here. So I hope this is clear to everybody, and the question also. The question is actually very logical, but anyways. Okay. Last question. What are the major reasons mammalian embryos develop within the parent organism as opposed to eggs in reptiles and other animals? Actually, an interesting question. So let me mark it as A, B, C, and D. A. Development of specialized features requires a constantly high nutrient supply. B. Better protection of embryo within the parent organism as opposed to in an egg. C. Mammals are warm-blooded animals and hence require a constant supply of blood from the parent during development. D. Mammals invest more resources into improving the survival of a few embryos, while reptiles invest more resources into increasing the number of embryos produced. Okay, these are your four options. You have 2 minutes. After 2 minutes, I'll switch the screen and then we'll discuss it. Think. Think what can be the answer. You have 2 minutes. Mm. 1 minute more left. Okay. Should we discuss this question then? Let us discuss it. Correct answer is all the options are correct. Why? First is development of specialized features requires a constantly high nutrient supply. Is it true or false? It is correct or not? Yes, it is correct. Why? See, mammalian embryos, it develops complex organs. You know, they develop large brains, they have an endothermic physiology. And the placenta, it provides a continuous flow of nutrients, oxygen, waste removal, which is far more effective than relying on a finite supply like yolk. A finite yolk is present in reptile eggs, but we do not need a finite supply. We because we are developing very complex organs, so there is even more requirement. There is there has to be continuous supply. So that is why when it is inside the body, there is a continuous supply which is provided by the placenta. So the placenta provides a continuous flow of nutrients, oxygen, there is waste removal, which is far more effective. So this is a correct option. Second is better protection of embryo within the parent organism as opposed to in it. Is it true or false? I mean, is it correct or not? Yes, it is correct. Why? See, developing inside the uterus, it protects the embryo from predators, desiccation, and also it protects from some kind of fluctuations from the environment, and which is not possible when there is a egg which is buried outside. So there is a protection. Okay, the environment temperature fluctuation is not there, environmental fluctuation is not there, predator fluctuation is not there. There's a better protection. So B is also a correct statement. Then, mammals are warm blooded animals and hence require a constant supply of blood from the parent during development. Is it correct? Yes. So, being endothermic, mammals maintain a constant body temperature, which provides the embryo with a stable environment for development, unlike reptiles, where external temperature affect that egg development. Okay. So, This is also a correct statement. And last statement is mammals invest more resources into improving the survival of a few embryos, while reptiles invest more resources into increasing the number of embryos produced. Yes, it is also a very correct statement. See, if you have studied, there is K selection, which happens in mammals, versus R selection, which happens in reptiles. So, mammals invest in fewer offsprings, but they provide greater parental care and higher survival chances. Okay, in mammals. Whereas reptiles, they lay many eggs with less parental care, accepting higher embryo mortality. Okay, is it clear to everybody? So, it is K selection versus R selection if you have studied. So, just for like in a basic knowledge, mammals they produce fewer offsprings, but even they have fewer offspring, but there is greater parental care which is being provided to the mammals. Whereas reptiles they lay many eggs, but the parental care is very less. So is it clear to everybody? I hope it is clear. Okay. So that's this is the last question. Yes. Okay. So with this we have just a minute. Yeah. So with this we have completed the questions of week 10. So now we have week 11 and week 12 lectures left with us. So we'll have those two, and we'll also have an extra session. And in that extra session, we'll actually kind of try to recapitulate what all we have studied. We'll try to revise it. Okay. And we'll see how it goes. So with this, we'll end today's class. Hope you were able to understand. If you have any problems, any queries, you can always contact me. You can always put your query in in my YouTube videos or mail me or whatever way you can find. So bye everybody. We'll meet next week.